Very warm welcome to all the participants who have joined us for this 18th session of BISMiS Live, our monthly scientific series on microbial taxonomy and systematics. The speaker for today is Professor Patrick Forterre. I'll give you a brief introduction about Patrick and his achievements. Patrick started working on DNA replication and DNA topology in bacteria. Inspired by Carl Woos's work, he turned out to study these mechanisms in archaea. And his first major contribution was the characterization of reverse gyrase, a novel type of DNA topoisomerase that introduces positive superturns into the DNA. And what Patrick could show that the reverse gyrase is the only protein that's essential for life at high temperature, which is actually a very major breakthrough in the science. Patrick defended his habilitation in 1985 at the University of Paris and became a professor at the University of Forterre, where he set up his own lab in 1989. In 1997, his team actually made another great discovery, and the discovery was that an enzyme that triggers meiotic recombination in eukaryotes, the SPO11, thanks to their work on archaeal DNA topoisomerases. In 2000, he made another great discovery where he identified the first DNA replication origin in the archaea. Patrick was then selected among the very few invited speakers at the meeting organized when Carl Woos received the Crafoord Prize in 2003 in Stockholm. In 2004, Patrick became the head of microbiology department at the Institute of Pasteur in Paris. He then set up in this institute a new lab while still running his lab in Orsay, which is actually it speaks volumes of Patrick's hard work and dedication that he can run 2 consecutive labs at 2 different places. He then invited David to work with him on archaeal viruses in Pasteur. They described several new viral families and fascinating new mechanisms of virus egress. Patrick thought that the importance of viruses was underestimated because they were confused with the virion. So he proposed the concept of a virocell or a virocell that posits that viruses transform the infected cell into a new type of cellular organism. In 2014, he received a 5-year ERC award to study virus-like vesicles in archaea. The interest of Patrick for archaea was early on boosted by their pivotal position in the universal tree of life, and he was greatly influenced and inspired by Carl Woos's work. And he promoted the name LUCA, which is stands for the last universal common ancestor. And very recently, he revisited with his co-workers, Violet and Morgan, the phylogenies suggesting that eukaryotes originated from Asgard Archaea. Their work supports the classical Woos's tree of life and suggests ancient gene exchanges between the Asgard and the ancestors of modern eukaryotes. Patrick is presently an honorary professor at the Institute of Pasteur and an emeritus professor at the University of Paris. I'm sure you'll learn a great deal of knowledge about the Asgard Archaea and the eukaryogenesis from Patrick's talk today, and this interest will be reflected in the question-answer session that we'll take at the end. And Patrick, it is my personal pleasure to be inviting you and hosting you for BISMiS Live. And with your talk as the first one in the series, we hope to have more sessions on eukaryogenesis and make our participants aware about this, very important origin of life, or evolution as we call it. So Patrick, the stage is all yours. Thank you.
Okay, thank you very much for this nice invitation. I'm very grateful to you to give me the opportunity to discuss about my personal view on the position of Asgard in the tree of life and the recent hypothesis that we put forward with 2 colleagues that many genetic changes take place between Asgard and proto-eukaryotes explaining some of the observations that we can make today. The topology of the tree of life is a very old issue. I recommend the reading of this chapter that I wrote with two former postdocs of mine, who now have permanent position at the Genoscope in France, the French Sequencing Center. We wrote a chapter about the tree of life in which we summarize the history of the debates between, especially the debates between the tree proposed by Carl Woese and the tree originally proposed by James Lakes. It's a very ancient story, since even the tree proposed by Jim Lake, in which the archaea were divided in two domains, subdomains, the archaea and the eocite, that back from 1984, in fact 5 years before Woese published his classical tree with a root position between bacteria on one side, archaea, eukarya on the other. Today, these three are usually called a three-domain and two-domain tree. And you see that the main difference is that in the three-domain tree, the archaea are monophyletic. In the two-domain tree, the archaea include the eukaryotes. So in fact, they are not monophyletic, except if we consider that we are ourselves archaea. In both trees, by the way, you have a clade formed by the archaea and eukaryote, both in the 3D or 2D tree. And I've used a few years ago to name this clade, which is normally the norm in the taxonomy to name a clade. I suggest you call it Archaea. Okay, at that time, nobody really used this name, but I will use it sometime in my presentation because it's sometimes useful. A very important point, difference between the two trees, is that in the case of the 3D tree, if you want to root the archaeal tree, you can use eukaryote as an outgroup. But in the case of the 2D tree or eocite tree, if you want to root the tree of archaea, you need to use bacteria as an outgroup. And this makes a big difference because the length of the branch between archaea and eukaryotes is much shorter than the length of the branch between bacteria and archaea. So, and the position of the root in the archaeal tree is important in taxonomy because it could determine the name of the higher taxa in the domain. Another point, another important difference between the two topologies is that in the 2D tree, all specific eukaryotic features, and there are a lot of them. I listed some of them here, like mRNA capping, of course, the nucleus, mitosis, meiosis, and so on. All these typical features should have appeared in the branch leading to eukaryote after the separation from archaea. Whereas in the 3D tree, the situation is more open. Some of them could have been already present before Luca, other in the common ancestral lineage between archaea and eukaryote, other in the specific eukaryotic branch. And some of them may have been lost either in both bacteria and archaea or only in archaea. The difference between the two topologies can be also important, is also important, if you want to reconstruct the proteome of Luca. In fact, in the 3D tree, some protein which are present only in bacteria and eukaryote could have been present in Luca and just lost in archaea. And it's theoretically possible to distinguish between the situation in which this protein was present in Luca, or if this bacterial protein was transferred, for example, through the mitochondria between bacteria and eukaryote. Theoretically, one can distinguish between these two scenarios based on branch size, which means that when you reconstruct the proteome of Luca, in that case, you have also to consider protein present in bacteria and eukaryote, but absent in archaea. In contrast, in the 2D tree, you don't have to take care of eukaryote, and this is the case for people who are at the moment try to do this reconstruction now usually, because the 2D tree is the most popular, they only consider bacteria in archaea, because in that case, protein which has only present in bacteria and eukarya are necessarily came from a transfer from bacteria via the mitochondrial route, for instance. So there are several important outcomes depending on the correct tree for all these aspects, and taxonomy is one of them. At the moment, most people consider that the two-domain tree is the correct one. This is a slide from a colleague in which he argued in favor of this view. And some people even consider that now the debate is closed after the discovery of Asgard. So Asgard were discovered by the group of Thijs Hetema in 2015. They published a very important paper in Nature. They published another paper in 2017. And in this tree, in fact, we can consider that the Asgard now correspond to the ancient earth proposed by Jim Lake. This is a drawing from a news and view in Nature about their paper. As you can see, the people were really like this paper because in fact they recover the ancient view that eukaryote came from prokaryote, and this gives a very nice classical view in evolution when you go from the simplest organisms to more complex organisms. This is really in contrast to the view of Carl Woese, who in fact this drawing from Carl Woese is a drawing from his review in 1981. This review was very influential for me. After reading it, I decided to work on archaeobacteria. At that time, he was speaking about archaeobacteria. And in this review, there was a figure in which he compared what he calls the older tree based on the physiological feature, which was popular at that time, in which eukaryote emerged by the fusion of several prokaryotes, and the new tree that he obtained based on the molecular sequences, 16S ribosomal RNA, in which the three lineage, the three domains emerged from what he called at that time the progenote. I was myself very intrigued by this 2015 paper. I was invited at the French radio to speak about this paper even before I could read it. And after reading it, I found that there was some problem. I will come back to that. And in 2017, with my co-workers, we published in PLOS Genetic a paper in which we criticized the conclusion of the Thijs Hetema group. And we consider that, in fact, at that time, Loki Archaea, which was the only Asgard named at that time, was not the sister group of eukaryote, but was in fact embedded within Archaea with eukaryote outside of Archaea. We also published an opinion paper in The Scientist. In 2018, a group of Thijs Hetema answered to our criticism in a PLOS Genetic formal comment, and a bit later we replied to their critical comment by our own critical comment. So it was a very lively debate. And in 2019, the group of Martin Eblé published another very influential paper in Nature, Ecology and Evolution, in which they say that they have definitive argument supporting the two-domain tree of life. Well, these are the two versions of the tree of life after the discovery of Asgard. For most people, so eukaryote emerged from within Asgard, which means, by the way, that if we believe this tree, we should consider that we are ourselves Asgard, taxonomically speaking. In the tree that we published in 2017, Asgard were branched between Euryarchaeota and Craniarchaeota, and eukaryote were outside. The branch of the archaeal domain in our publication was located between a superphylum that we call bats for Bathyarchaeota, Algiarchaeota, and Chromarchaeota, and all the other archaea. This is important for the taxonomic system because, for example, in the recent taxonomic proposal by Rinke and co-worker, if this route is correct, one of the major proposal cannot be, is not supported taxonomically speaking. Recently, many new lineages of Asgard has been discovered by Chinese colleague. First paper was published in Nature, and a second paper was published a bit later on. And Chinese colleagues suggest me to write a review about this new discovery in the new journal MLife. And so with Morgan Gaia and Violette d'Acunha, my two long time now co-worker, we wrote this paper expanding Asgard Archaea and the elusive relationship with eukarya. And I highly recommend, if you are interested by the topic, to read this paper. I will make several times I will mention this paper. Yes, for example, this is a figure taken from this paper in which we summarize the phylogeny of Asgard. There are 18 Asgard phylum formally proposed at that time. This tree was rooted, I don't know, more or less according to the publication paper which have been published on the topics. And the two red circles indicate the two different positions of eukaryote which have been found by this author who obtained 2D tree when they built universal tree based as usual on the concatenation of universal proteins. So I want to come back to the 2015 papers and explain why I became skeptical about the position of Asgard in the first tree published in this paper. This tree was based on the concatenation of 36 universal protein, and three Asgard were the three mags of metagenomic associated genome, three mags were known at that time, which were called Loki 1, Loki 2, and Loki 3. And now, after the discovery of many new lineage and new phylogenetic analysis, two of this lineage correspond to Archaeota and Oda Archaeota. Maybe you can see on this figure that, for example, Loki Archaea, I mentioned Loki Archaeota, which is a phylum, or Loki Archaeales, which is an order. This is because there is a controversy at the moment about the taxonomic situation of Asgard. Most people used to consider that Asgard forms a superphylum with a lot of internal phylums, and others consider that Asgard should be considered to be a phylum by itself, and in that case, this lineage are no more phylum, but order or class and so on. I will not enter into these debates today, so sometime I will use Loki or Oda, for example, for Arde Archaeota or Arde Archaeales and so on, or Emdal and Karya for Karya Archaeota. So it turned out that Loki 1 was a Loki, Loki 2 was a Karya, and Loki 3 was a Oda. And in this tree, eukaryotes were embedded within Asgard and sister group of Loki 3, so sister group of Oda. I will come back to this point later. The first thing which struck me when I look at the tree was that two well-known fast-evolving archaeon, Metalopyrus candleri, was located at the base of the tree, which was not normal because normally Metalopyrus candleri is closely related to Metanobacteriales within Euryarchaeota. So this indicated that there was a long branch attraction between Metanopyrus candleri and bacteria. And Dipan, which correspond to also fast-evolving organisms, were located close between two groups of Euryarchaeota. So there were problems in these trees. I don't recover the normal phylogeny of Archaea, which means that despite the fact that they claim using very evolved, sophisticated method to build the tree, still the tree had some problem, especially long branch attraction problem. Thus, I decided to have a look by myself at this protein of these new organisms. I looked just by chance to a ribosomal protein, the S5, and I did myself show a rapid alignment of several sequences from this protein from the 3 domain of life. And when I look at the sequence of the Loki protein, I could not find any similarity, specific similarity with the eukaryotes. And I found that indel, like deletion or insertion in this Loki sequence, they were very, very typical of archaeal indel. So I could not see why they found finally the Loki close to eukaryotes. So I decided we should go further, and together with Violette, Dacunia, and Morgan, especially Violette, rebuilt the 36 tree of universal protein using the same data set of Etema and co-worker. And I think it's very, very important always to look at individual tree. We found one Woos tree and 35 earseed tree 2D. So not surprising that the final concatenation given an earside tree. But we noticed that the position of the tree Asgard were variable, very variable from one tree to another. Asgard were often paraphyletic, with sometimes some Asgard close to eukaryote and other embedded within Archaea. And the support for the branches were always very, very low, which is not surprising for single tree, with some exception. I will come back to that. When we look, for example, at the position of Crenarchaeota or Euryarchaeota, we don't get the same, we don't make the same observation. They were often monophyletic. So this, the fact that the Asgard were very often paraphyletic in this tree was not in the normal situation for lineage belonging to the same phylum. But surprisingly, among these 36 trees, we found one of them, which was exactly similar to the 36 protein tree published in Nature. So it was very amazing. One of the 36 trees was exactly the same with, as you see here, the three Loki close sister group to eukaryote and eukaryote sister group to Loki 3. So it was really, really surprising. So we looked more carefully at the sequence of this protein, the elongation factor named EF2 in eukaryotes, EFG in bacteria. An amazing thing is that there was one difference between the two trees. The position of Metanoparus condleri, which was not correct in the tree published in Nature, was correct in our tree. And the only difference is that in the tree published in Nature, they did a Bayesian, they use a Bayesian model, sophisticated. And in our case, for rapidity reason, because we want to go fast to have this 36 tree, we did, and also because Bayesian analysis is not good for single tree, but better for concatenation, we did a classical ML tree, and with a classical ML tree, we get the good position of Metanoparus condleri, which is interesting for me. So we look more carefully at the sequence of this elongation factor EF2, and we found several for eukaryotic-like insertion, very typical in the sequence of the ODA, let's say ODA. One of them was present also in Loki and Carrier, but three were specific of ODA, which recapitulate, by the way, the relative position of these three Asgard compared to eukaryotes. This is one of the three specific indel present in the ODA. It's a rather long indel, and you see here that this indel, that you can see here corresponding to Loki 3, ODA, this indel, it's about 10 or 11, I don't remember, amino acid, is absent in other Asgard, absent in Archaea, and present in eukaryote, and more specifically here, not in UF2, but in a paralog of UF2. So at that time, we think that really this should be a kind of contamination that probably there, when they reconstructed the mag of this Asgard, they incorporate some sequence coming from eukaryote living in the same environment. It was a contamination. We changed our mind later on because, in fact, this indel was recovered in another mag, independently obtained, I think in China or I don't know. yes, I think in China, which means that in fact there are now two mags of ODA and they both contain this indel. So it was difficult to imagine that by chance these two mags were coming from different labs, from different regions of the world, were contaminated by the same eukaryotic sequence. So I will come back to this point because now we think that this is an indication of this transfer between proto-eukaryote and Asgard. What we did after we observed this similarity between EFG, between eukaryote and Asgard, we remove EFG from the data set used by the group of ETEMA, and we built a tree based on the concatenation of the 35 of the protein. And the result was dramatic. Removing a single protein out, one protein out of 36, the topology of the tree changed quite a lot, especially eukaryote move. So we always get the 2D tree, but eukaryote moved far from Asgard. They were now branching within Archaea, I think. So this really shows that a single protein can dramatically change the result of a concatenation. I should say that from that time now, the group of Thais ETEMA and other, they don't use anymore EF2 in their analysis. In this tree, we have the DPAN, which branch at the base of the Archaea, which are fast-evolving organisms. And the DPAN is a superphylum which include many small Archaea living in symbiosis with other Archaea. They have been named after Diapherotretes, Parvarchaeota, Henning-Marchaeota, Nanoarchaeota, Nanohaloarchaeota. There are more now, like Woos-Archaeota, Pace-Archaeota, and so on, Undine-Archaeota. And we noticed in our 36 trees that they were also often paraphyletic, like the Asgard, and the support for their position was always low to very low. These organisms are also very fast evolving. To illustrate this point here, you have a short region of the ribosomal protein L22. This protein is very well conserved between eukaryote at the top in blue, bacteria at the bottom in green. And most Archaea, you see there is not a single amino acid, there is no insertion of deletion, except here you can see in DPAN, I would say even in Methenopyrus, which is a fast-evolving archaeon, or in Corarchaeon, which is another fast-evolving archaeon, there is no indel. But if you look at DPAN, you have many deletion or even sometimes insertion. So this indicates that protein from DPAN evolve very rapidly, and this will be, of course, a problem for phylogenetic reconstruction. You all know probably about this tree of life, which have been published in Nature Microbiology as a new view of the tree of life, in which DPAN are at the base of the Archaea, similar to so-called CPR, candidate phylum radiation, which are also very small bacteria living in symbiotic association with other organisms, and which in this tree also branch at the base of bacteria. Obviously, this is the result of long branch attraction of DPAN by bacteria, attraction of CPR by Archaea. In the case of CPR, now more recent work, in particular by Martin Gutierrez and Frank Elward, have shown that, in fact, when they use more sophisticated method to build the tree and to select universal protein, when they built a tree of bacteria using Archaea as an outgroup, now this DPAN called Patacibacteria, they branch within bacteria, sister group to a chlorhexane. This has been found by other authors. So in the case of DPAN, it's here in their work, DPAN still branch at the base of Archaea, but this is also controversial. So what we decide to do is to go back to the initial data set of Taisetema and colleague, but remove fast-evolving species. So we remove all DPAN, we remove Metanoparus candleri, we remove Archaeota to try to avoid all this problem of long branch attraction that can be triggered by fast-evolving organisms. And to our surprise and pleasure for me, because Violet and Morgan were neutral, considering the 2D, 3D, for me, I should say that I always prefer the 3D. I have no time to explain why. There are many other reasons for me. But to my surprise, this 2D tree was transformed into a 3D tree. So eukaryotes were now outside of the archaea, and Asgard were at the base of the archaea. So this clearly indicated that the 2D tree published in Nature was due to the presence of EF2 and fast-evolving species, EF2 in the protein dataset, and fast-evolving species in the species dataset. And our supposition was that probably, in fact, when you have this kind of analysis, which includes fast-evolving organisms, probably the normal tree here on the left, which is a 3D tree, was transformed into a 2D tree by the attraction of DPAN by bacteria, maybe the attraction of Archaeota by DPAN, which brings the eukaryote within the archaea and close to the Asgard. The fact that Archaeota can be also attracted by bacteria is illustrated by a work published a few years ago by Simonetta Gribaldo and co-worker, in which they produce a tree of, they root the archaeal tree using bacteria as an outgroup in the framework of a 2D topology. I tell you, if you have a 2D topology, you need to use bacteria as an outgroup. In that case, they obtain the root of the archaeal tree within Archaeota. They divide the Archaeota in between two major groups. So this, for me, because the length here is very long between bacteria and archaea, this is not the correct route for me of the archaea, but this testifies for the fact that Archaeota probably are the most fast-evolving archaea once you have removed, of course, DPAN. And in this particular work, they have not included the DPAN in their analysis. So this could explain the attraction which could be responsible for the to go from a 2D tree to from a 3D tree to a 2D tree. Okay, the 35 protein tree that we obtained was not robust. We have Asgard at the base of the archaea. The tree was the bootstrap value or the value were not very good, and the branch defining the monophyly of archaea is very short. I should say that it is this tree which has been criticized by Embley and co-worker in their 2019 paper. They focus their criticism of our work on this particular tree, and using a new evolutionary model, starting from this tree, they obtain a 2D tree. And they were very happy because they are long-time supporter of 2D tree. And you see, as I tell you, it's very easy to eliminate this branch, depending on the model you are using. But we went a bit further when we reanalyzed the 36 tree after removing fast-evolving species. First of all, we noticed that we increase the number of whose tree. We get 11 whose tree against 1. And we did some statistical tests to test the robustness of this tree. And we found that out of the 11 trees, 6 were very strong. And from the 24 2D tree, 11 were very strong. So what we did, we concatenated independently the host protein and the eocyte protein. So the protein producing a host tree and protein producing an eocyte tree. And of course, this is not surprising. Putting together a protein giving host tree, we obtain a very nice 3D tree with Asgard within Archaea. And after the concatenation of the protein giving 2D3, we obtain a 2D3, the classical 2D3 obtained first in 2015. But the interesting point here is that in this case, we use the same model for the 2D3, Bayesian model with the most sophisticated model known at that time. So it means that, in fact, with the same very elaborated model, you can obtain completely different topology depending on the protein you are using to make the concatenation. This is a very important point. Also very important, we look at if there was a correlation between the size of the protein and the fact that they produce a 2D or a 3D3. And clearly there was a correlation. In red, you have protein producing a 3D3, and they are classified here according to the length, number of amino acids. Here you have larger protein, here you have smaller protein on the left, larger protein on the right. You see that protein producing 3D3 tend to be larger, and protein producing 2D3 tend to be lower. This is striking. So these two, the two largest universal protein are the two large subunits of the RNA polymerase. And here, the largest protein producing a 2D3 is EF2, EFG, which I mentioned previously is a problem. So we should have removed this particular column. So this for us explains a lot, because it could suggest that in fact you need large protein to recover the short archaeal branch. If you have smaller protein, in fact, the signal of this branch disappear in the general noise of the analysis. And in fact, to come back to the more recent works done by the two Chinese group, Liu and co-worker, Xi and co-worker, in fact, several proteins which give a Wu's tree in the original data set, and that we recover in analysis, are missing in the data set, you see. In fact, here you have in red the protein which give 3D3 with strong support, with low support, and gray protein giving 2D3, strong support like AFG, low support. And you see that some of the red protein here are missing in Liu, Xi, and so on and so on. So, and their data set contain also many of the small protein which produce 2D3 like in the original one. So at the time in 2015, 17, let's say, after we obtain this result, we decide to use the two large subunits of the RNA polymerase to build a universal tree, reasoning that this could be the best universal tree we could obtain. So this is indicated here. We built a new universal tree based on the concatenation of the two large universal protein. And we also make another change compared to the original paper. The original species dataset of Spong and co-worker contain 10 bacteria, 10 eukaryotes, and 87 archaea. And that it has been shown later on by other colleagues that such imbalance in the representation of the different organisms in each domain can create problems. So even at that time, we use a new data set with 39 archaea, 39 bacteria, 39 eukaryotes without fast evolving species. And more recent work by Frank Elward and Carolina Martinez-Gutierrez, in fact, prove in my mind that we were right to choose an RNA polymerase, because they found that when they look at the different universal protein and the quality of the signal produced by this protein, they conclude that RNA polymerase large subunits have the highest phylogenetic signal and outperform a set of 41 conserved archaeal and bacterial markers despite having a shorter overall length. That is a very important point. Even if putting together the 41 protein, the length is even larger than the putting together the two large RNA polymerase subunits, the phylogenetic signal remains better in the RNA polymerase. And in contrast, they notice that ribosomal protein tend to carry low phylogenetic signal. And this is a very important point because now most people tend to use ribosomal protein in their analysis, especially ribosomal protein. So when we produce this RNA polymerase 3, we obtain a nice, strongly support using Bayesian model, most sophisticated model. We obtain a very well-supported 3D3 with Asgard in the middle of Archaea as sister group of Archaea and eukaryote very well separated from Archaea. But notice that the branch length specific to Archaea still remain rather small. Interestingly, we obtain exactly the same topology than we obtained previously when we concatenate the 6 WOOS protein, giving strong support for the WOOS3, in which there was only one of the 2 RNA polymerase subunits. So we think that this topology is probably good. And in this topology, you see that the root of the Archaea tree is between Tomarchaeota and the other. So now when we had Archaea and Archaea, they came with Tomarchaeota, so we call this bat phylum. And this is important for taxonomic purpose because if this is correct, some of the recent proposals for the taxonomy of Archaea are not valid if you take a cladistic approach for taxonomy. In their paper in 2019, Emble and co-workers, they in the supplementary material, in fact, they work again on a RNA polymerase 3. They use their new model to try to obtain a 2D3 with RNA polymerase, but despite this new model, they still obtain a 3D3. They put this in the supplementary material, and then they do amino acid recoding, and only after amino acid recoding, they obtain a 2D3 with the RNA polymerase. And they say this is another argument, finally, the 2D3 is correct. But in fact, for me, this is really misleading because amino acid recoding is not neutral. And this has been shown, for example, yes, not neutral because it reduces the signal. And I show you that the signal is very important in that case because of the small branch of the Archaea. And this has been shown in a recent paper by Hernandez and Ryan. They conclude that 6-state amino acid recoding is not an effective strategy to offset compositional heterogeneity and saturation in phylogenetic analysis. Clearly, recoding reduces signal, and I suppose that recoding eliminates the signal that correspond to the archaeal branch. And in fact, when you look at the tree obtained by Martin Emble and co-worker after amino acid recoding, you see that the tree of Archaea is very unusual. Crenarchaea are paraphyletic, and eukaryote branch within Crenarchaeota. And you have many basal branches in eukaryotes. So clearly the tree is very bad. So it cannot be considered as a valid criticism of the tree which give the WOOS topology without amino acid recoding. Okay, so last question about the topology itself. It could be argued that possibly the 3D3 is due to the attraction of the long branch of eukaryote by the long branch of bacteria compared to the shorter branch of Asgard. This was a really valid criticism, but we think we have answered to it when we had the 3 eukaryotic RNA polymerase in the tree. When you had the 3 RNA polymerase, usually people use eukaryotic RNA polymerase 2 in this kind of analysis. When you had pol 1 and pol 3, in that case, here you have you have 3 times the last eukaryotic common ancestor, and here you have the last eukaryote, which still has only 1 RNA polymerase. So the branch became much shorter, but then we still get a 3-day tree. So in this MLife paper, we give some indication to what could be done now to continue and to try to get more convincing data in favor of one of the other 3. One important point would be to work more on the protein data set, because here you have you see the different columns correspond to the different protein used by different groups when they publish universal 3. And you see that all groups, they use different sets of protein. Nobody use a complete set. And even if you use a complete set, you have to be careful to distinguish between the size and so on and so on. Some of these proteins have parallax. It's a huge amount of work, but I think it's a very important work which has to be done in the future. And you can read all these suggestions in the MLife paper. So now I want the second part of my talk, which will be a bit shorter, but which corresponds to the idea of this gene transfer. If the 3D model is correct, what I think, how to explain that there are so many eukaryotic signature proteins, so-called OSP, in Asgard. This was one of the very strong arguments for people who claim that eukaryote can emerge within Asgard, is the presence of this OSP, such as actin, tubulin, very similar to the eukaryotic homologue, or else actin-related protein, which are called ARP, which are also present in some Asgard, and there are many others, like the trafficking machinery, escort machinery, and so on. So how to explain the abundance of ESP in Asgard in a 3D model? One observation which has been made early on, and which was already visible in the paper published by the group of ETEMA in 2017, is that the ESP exhibit a patchwork distribution among Asgard. There is very few universal OSP among Asgard. Usually they are limited to 1 or 2 phylum. At that time, there were not so many phylum, but for example, you can see here, tubulin was present only in Odin Archaeota, and here a protein looking but very different, but looking like the DNA polymerase epsilon in eukaryote was also present only in 1 Asgard. The discovery of new Asgard by the 2 Chinese group expand even more, by the way, the diversity of OSP. This is a picture taken from the paper of Liu and co-worker. You have here at the top the different phylum of Asgard. Here the presence or not of many OSP which have been discovered now. And you see that very few, in fact, actin and some is one of the only, if you include actin-related protein, there is 1 actin which is present in all Asgard, but others are present usually in 1 or 2 phylum, and some even only in 1 phylum. This is the case of tubulin, for instance. Tubulin still is present only in Odin Archaeota. So tubulin now is present in 1 out of 18 Asgard lineage, which is really surprising. It's very difficult to explain this in the classical 3D model. Interestingly, the same situation has been observed for tubulin in bacteria. Tubulin is also present in a few bacterial species. And in that case, the obvious answer by the author is that it was an horizontal gene transfer between proto-eukaryote and bacteria. But of course, this explanation has never been proposed for Asgard OSP because there are such empathy for the 2D domain today, and so many people like the idea that eukaryote came from Asgard. But when I look at the 3 published by the group of ETEMA, in fact, in the 2017 paper, they published a phylogeny in which they include the tubulin of Odin Archaea. You see that, in fact, the tubulin of Odin Archaea branch very close to the eukaryotic one, and very far from the tubulin found in other archaea, like in tubulin found in some Tomarchiota. And if you have in the same tree, you have the tubulin of bacteria, you see that in that case, they have been suggested to be transferred from proto-eukaryote. Why not propose the same explanation in that case? And why we say proto-eukaryote? Because you have this clade corresponding to eukaryote, and at the base of this clade, you have the last universal, the last common ancestor of modern eukaryote, the last common ancestor of modern eukaryote. I drink a bit of tea. And in fact, the hypothesis of horizontal gene transfer can easily explain the patchy distribution of ESP in Asgard. If this transfer occurs very early on at the base of all Asgard, which may be the case of some actin, then it will be present in all Asgard. If the transfer takes place later on, during the diversification of Asgard, then the protein will be present only in the 1, 2, 3, several Asgard lineage. This hypothesis has some implication. If it's true, it suggests that the diversification of Asgard takes place before the time of the last eukaryotic common ancestor, which means that the evolution of proto-eukaryote takes quite a long time. It means also, but I have not done that yet, that possibly it could be possible to reconstruct the phylogeny of Asgard also based on the relative timing of these transfer. There is also a strong argument from phylogenetic analysis for the fact that actin Asgard was transferred from proto-eukaryote. This is a tree in which you have the classical archaeal actin, which are present in several archaeal lineage, in Archaeota, in Archaeota, and others. And here you have Asgard lineage in purple, which branch very, very close to their eukaryotic counterpart. Here we have found also as actin in Archaeota, which means that sometimes you find ESP not only in Asgard, but in other groups which are unrelated to Asgard. We call it actin. We call Asgard actin the different group of actin found in Asgard, and they are very, very divergent clade, but all grouped tightly together with the eukaryotic one. And in blue here in this tree, you have the clade of Asgard actin R. I will show you now a more detailed tree of the world of actin that we obtained more recently. It's a work by Violette d'Acunha, mainly, who, in fact, we discover that some giant viruses encode and also encode actin. We call them viractin. We found 4 clades of this viractin 1, 2, 3, 4. So we built a new tree. This is published now in MBE. We built a new tree in which we include this viractin, the Asgard actin, the actin, and the different group of eukaryotic actin and actin-related protein. And the tree is rooted with actin, so the classical archaeal actin. And I would suggest that these classical archaeal actin are the real archaeal actin, the one which diverged from eukaryotic actin when the lineage leading to archaea and eukaryote diverged. And you see that in the purple here, the Asgard actin, there are several groups too, they branch in between the last eukaryotic common ancestor in yellow. Here, at the base of each of these clade of ARPs, which are present in all modern eukaryotes, they should have been, their ancestors should have been present in LECA. And you see that here you have LECA, here you have LECA, and here you have at the base the ancestor of this clade of Asgard actin. So it's a strong phylogenetic argument to say that actin that we found now present in Asgard have been transferred from proto-eukaryote. So we wonder, is it possible to explain the odd phylogenetic pattern of some Asgard universal protein by this hypothesis of horizontal gene transfer? I tell you before, when we look at the individual statistics tree of the first Nature paper, we found that in some cases, we found that generally speaking, the position of Asgard moved from one tree to another. Sometimes we found some Asgard very close to eukaryote and other far from eukaryote. So first, I will show you two cases that we identified. The first, of course, I come back to this EF2 protein in which we found this specific eukaryotic-like indel, which is present in an archaeon which belongs to the Oda lineage. I tell you that now there are two mags from this Oda lineage. And in fact, when I look at a paper by the group of Tai Setema in which they discuss the evolutionary history of EF2, I notice that, in fact, they have a strong argument in favor of our hypothesis, even if they don't mention it. In fact, in this tree, in their tree, you have Asgard divided in two groups, huge group with most Asgard present between Eukaryote and Archaea, as in our analysis, and then two Asgard, two other, the two which contain the indel, which are close to eukaryote. I focus here. So it's quite obvious that this F2 with a eukaryotic-like indel presence in these two Asgard came from gene transfer from eukaryote. This for me is now really the best explanation. But we found another case, very interesting, when we look at the data set of Menli and colleagues, one of the last two Chinese paper, we focus on a protein which is called K1 in YGGD. It's a protein we know very well because when I ran my laboratory in Orsay, we did a lot of biochemistry on this protein. We purified these different complexes and so on. And this is a universal protein which has different name depending of the domain. It's called K1 in eukaryote and archaea and YGGD in bacteria. So when we look at the tree of the when we reconstruct the tree using the data set of Menli, we obtain a 2D tree, but you see that in this 2D tree, we have 10 phylum of Asgard forming a clade, and two other phylum of Asgard, Emdal and Carya, sister group of eukaryote, and far from the other one, and grouping with Eukaryote. You notice that these three, they have at the base of Archaea the dipan. And I tell you that we don't like having a dipan in this kind of analysis because they evolve so rapidly that they can produce artifacts. So we remove the dipan, we run the tree after removing the dipan, and we obtain this new tree in which now the clade grouping eukaryote and these two Asgard were outside of the Archaea. So obviously, it means that probably these 10 phylum which form a clade correspond to the right position of the Asgard within Archaea, and these two clade replace, in fact, their ancestral protein, K1 YGGD protein, by a protein from proto-eukaryote. And by the way, if you remove these two phylum of Emdal from the tree, then you obtain a very nice 3D tree, meaning that, in fact, this transfer from proto-eukaryote to Asgard can also mislead the analysis and produce 2D tree when normally you should have had a 3D tree. And we notice that Asgard, in that case, branch between Eukaryote and TAC and Kren Tom, as in the RNA polymerase tree, or as in the tree that we built using the 6 protein producing strong roots tree. What is interesting for me is that the horizontal gene transfer hypothesis between proto-eukaryote and Asgard suggests also new hypotheses. I forget to say that, by the way, this hypothesis is also valid if the tree is 2D. It's valid in both 2D and 3D, but in the case of 3D, it explains well the presence of this OSP in Asgard. It suggests that proto-eukaryotes probably lived in a biotopes similar to those in which modern Asgard are thrilling. which means that when you collect modern Asgard, possibly you collect sample in environment very similar to those in which your eukaryotic ancestors were thriving. It means also that Asgard Archaea give us access to ancestral state of this protein in proto-eukaryote. So we could imagine to reconstruct the pathway from the last archaeal eukaryotic common ancestor to LECA. Maybe if I come back to this tree, for example. Oh, yes. This one. Yes, you see that in that case, you have a lot of nodes in this tree, a lot of nodes between LECA, the last eukaryotic common ancestor, and here you would have the last common ancestor of archaea and eukaryote. You have many nodes, and phylogeneticists, they know how to reconstruct protein sequence of this node. So we could imagine to reconstruct the ancestral protein sequence of proto-eukaryote and try maybe to reconstruct the pathway from LARCA to LECA. Finally, if we remind that the first isolated Asgard live in close association with a methanogen and the bacterium, so it's an obligatory symbiont, the Candidatus prometheoarcheum centrophicum, which is the only Asgard which has been cultivated until now. When you read the paper of the Imashi and co-worker, doing the metabolic analysis of all Asgard, they suggest that in fact, they say we provide evidence that Asgard Archaea are dependent on symbiotic interaction for both catabolism and anabolism. And this reminds me that the position of Asgard in individual trees were variable like the position of Dipan. So I suspect that Asgard Archaea, maybe like Dipan, are rather fast evolving species, and they could be also obligatory symbiont, most of them. This would explain very well why they are so difficult to cultivate, why nobody yet succeed to cultivate more than one archaeon, despite the fact that you know that if you succeed to publish an Asgard, you will publish in Nature, so many labs should be trying. So this is an hypothesis, but it may be that all Asgard could be dependent of symbiotic relationship, explaining the rather fast evolving phenotype of the protein. And in that case, why we could imagine that maybe the reason why there were so many horizontal gene transfer between proto-eukaryote and Asgard could be due that some ancient Asgard live in close association with proto-eukaryote as ectosymbiont of proto-eukaryote. And this leads to a very interesting idea, according to me, that maybe, even today, some Asgard possibly still live as ectosymbiont of modern eukaryote, modern protists. And I think it could be a nice research project to look for them in the environment. So I will stop on this proposal and conclude that in my mind, the present data are strongly in favor of the classical Woos tree, even if most other scientists still need to be convinced. I predict that the controversy will continue, and it will be difficult to convince some scientists because the 2D tree fit too well with the paradigm that eukaryotes are more or less superior organisms, with Homo sapiens superior eukaryote, which emerged from modern prokaryote meaning before karyote. And I am a bit sad about that because, in fact, with this two-domain model now, in which bacteria and archaea produce eukaryote, we come back to the pre-Woos pre-Woos situation in which people at that time, like Lynn Margulis, already imagined that maybe prokaryotes came from the eukaryotes came for the merging of different lineage of eukaryotes. So thank you very much for this invitation again and the opportunity to present this data and these hypotheses. And I'm ready for discussion. I hope I've not been too long.
Not at all, Patrick. Thank you so much for this excellent talk. I'm happy to take questions, and there are very many questions that have come. I'm taking them based on the number of votes each question has received, so that the more popular questions are taken first. So the first one comes from an attendee by the name Matrix, and the question is, Thank you very much for the excellent talk. I have three specific questions. The first is, Thomas Cavalier Smith put the root of common ancestor of Archaea and eukaryote in the PVC group of bacteria by using ribosomal proteins and regards Archaea as a side branch of evolution. How do you view these points?
Yeah, I think I'm strongly against this view. I had no time here to speak about too much about Luca, but for me, the root of the tree cannot be localized. The root of the universal tree cannot be localized based on phylogenetic analysis because they are too divergent. When you look at duplicated proteins, they are too divergent. For me, I think the best things to look at is to look at the distribution of ribosomal protein in modern organisms. In modern organisms, you have 34 ribosomal proteins which are common to the tree domain, 33 which are common to archaea and eukaryotes, and not a single one common to bacteria eukaryote or to bacteria archaea. Which means that if you have a LUCA, for example, which is present in bacteria you have 23 unique protein, ribosomal protein. As I said, 33 specific to archaea eukaryote. So if we start with a LUCA within bacteria, which means that LUCA had these 23 ribosomal proteins specific of bacteria. And then you should imagine that it replaced them, it lost them, and replaced them by the 33 protein specific of archaea and eukaryote. And for me, this is very unlikely, and I don't see any selection pressure to replace these 23 ribosomal proteins. The ribosome of bacteria is working very well by this other protein. It's the same if you put the root in my, when I was rather young, I used to prefer the root of the tree in the eukaryotic branch. But now I abandon this idea because in that case, it's the opposite. You start with a ribosome which includes the 33 protein of common to archaea and eukaryote. You should lose them in the bacterial branch and replace them by the 23 proteins specific of bacteria. This seems for me very, very unlikely. And you can make the same reasoning for other mechanisms like transcription and so on, replication. So I think there is a strong argument from comparative molecular biology. It's easier to imagine that LUCA has only the 34 universal protein, and then you add 23 protein in the lineage leading to bacteria, and you add 33 protein in the lineage common to archaea and eukaryote. And this has important consequences because it means that probably LUCA was less complex than modern prokaryote. It was simpler than archaea bacteria, a bit like Carl Wood and George Fox predicts the progenote story. It has smaller ribosome with only about 40, maybe 35, 40 protein, which doesn't mean that it was not able to make nice protein because we know that among universal protein, you have some, for example, tRNA modification factor, which are important for faithful translation. For instance, the Ki1 protein I mentioned, this Ki1 protein associated to this other universal protein. It has been shown in mitochondria that it can introduce this specific tRNA modification, which is very, very important for faithful translation, suggesting that LUCA was already able to do that.
Okay. Yeah. The second question that is from Matrix is that, do you at least partially abandon the thermoreduction hypothesis since you put forward the proto-eukaryotic horizontal gene transfer hypothesis?
No.
Okay.
Thank you for this question. Thank you for this question. I don't have a slide here about this thermoreduction hypothesis, but it's related to the story of LUCA. In fact, Yes. Maybe I can just come back to one of the universal three at the beginning. Yes. Yes. In fact, if Luca only had 34 ribosomal proteins, so if Luca was, as I say, simpler than modern archaea and bacteria, it means that the phenotype of archaea and bacteria, what we can call the prokaryotic phenotype, they have small size, rather small genome. There are exceptions, but usually they have small sizes, small compact genome, and so on. This means that this phenotype take place independently in bacteria and archaea. To illustrate this point, when I did a seminar on Luca, I have a Tasmanian wolf and a wolf, which belong to different branch of mammals, but which look very similar when you look at the shape. So bacteria and archaea could be very similar in shape, even if they came from an organism which was, if they belong to different branch and come from a common ancestor which was very different from them. And one of the explanations for the modern phenotype of bacteria and archaea could still be the adaptation to high temperature. It has been shown by the work of Manolo Gui in France, in Lyon. They have shown when they reconstruct the ribosomal RNA of the last common ancestor, they found that Luca was mesophysed, but they found that the last bacterial common ancestor and the last archaeal common ancestor both were thermophiled, even hyperthermophile for the archaea. So you have to understand how, from a Luca living at rather low temperature, you arrive at a common ancestor of bacteria, common ancestor of archaea, which live at high temperature. So this could be, for me, an argument in favor of this thermoreduction hypothesis, suggesting that at some point, maybe, for example, DNA, the replacement of RNA by DNA, which is much more stable, was induced by this kind of thing. This is another seminar.
Okay, yeah.
That's a very good question. Thank you.
Yeah. The last question from Matrix is actually an opinion he's asking of you. And the last question is that if Carl was still alive, when do you think he could receive his Nobel Prize?
Why she? Why? Yes, I think that Carl Wood should have get the Nobel Prize because it's a Nobel Prize of medicine. But in fact, his work, the idea to use ribosomal RNA and macromolecular sequences to determine the relationship between organisms, and also after that to identify organisms. So the work of Carl Wood and his postdoc Norman Pace opened the way to microbial ecology and modern metagenomics, and this had a tremendous importance also in medicine. So I think that Carl Wood should have deserved the Nobel Prize. He gets the Crawford Prize, which is very important too. It's the same amount of money. He received it from the hand of the King of Sweden, so it was nice. But he would have also deserved the Nobel Prize, because I used to say that the difference between the Crawford Prize and the Nobel Prize are very similar, as I say. The only difference is that for the Nobel Prize, you have many journalists and coverage by the press, and the Crawford Prize, no journalists, no coverage at all. So it was very unfortunate that Carl Wood get the Crawford Prize only, not the Nobel Prize. And I was also rather sad about that, because I was very happy recently when Svante Pabo got the Nobel Prize for the sequencing of the Neanderthal genome, which it was not obvious for a Nobel Prize of medicine. So if Svante Pabo gets the Nobel Prize for the sequencing of the Neanderthal genome, which is very nice because this sequencing was a daunting task, I think Carl Wood should have also got the Nobel Prize. No reason why he doesn't get it, except that there was always people, especially in the USA, fighting against this idea.
Yeah. Okay. The next question comes from Maggie Low-Wetter, and the question is, thank you very much for the presentation and much appreciated insight into this important topic of 3D versus 2D. You showed that two large ribosomal proteins, RPO proteins, give the highest phylogenetic signal, whereas R proteins tend to carry low phylogenetic signals. RPO catalyzes the process of transcription, whereas R proteins are involved in the process of translation. With RPO giving higher phylogenetic signals than R proteins, does it speak in favor of the RNA world?
No, I don't think so. I think they give a better phylogenetic signal mainly because they are large. And maybe they are more constrained because they are enzymatic. They have enzymatic activity, which means that probably there are more constraints on their sequence than in the case of ribosomal protein. But I don't think this give any indication. I strongly believe that DNA genome were preceded by RNA genome, and there are several arguments suggesting that LUCA itself could have still had an RNA genome. Maybe DNA was introduced later on during the lineage of proto-bacteria, proto-archaea, eukaryote, and so on. But I don't think that. I think the fact that RNA polymerase has a higher phylogenetic signal is really related to its catalytic activity.
Okay, thank you. The next question is from Devananda, and the question is, what's the relationship of Asgard with halophiles, methanogens, and thermoacidophiles, and whether the Asgard are closer to thermoacidophiles than halophiles?
The Asgard, for me, are a bit closer from thermoacidophiles, but they really branch. They really branch between Euryarchaeota and all the other archaea. The topology of the archaeal tree, in my opinion, is not completely resolved, especially the position of the Stygia, like Asgard Archaeota, and the position of Dipan, the position of several new groups which have been recently discovered, and the Asgard. I'm working on that using Indel in some universal protein, and it's a complicated story. The simplest thing to say is that they are between Euryarchaeota and Crenarchaeota, Tomarchaeota, that's for sure. So they are really separated from these groups, but they are close from other groups like Dipan, Hades, and so on.
Okay, yeah, thank you. Some of our participants are actually beginners in the field of microbiology and phylogenetics. So some of the questions are, very much related to what method or algorithm to use for phylogenetics. And this question is very similar. The question is from an anonymous attendee, and the question is there any guideline as to when to use maximum likelihood and when to use Bayesian inference methods? For concatenated sequences, do you always recommend Bayesian inference?
Okay, Bayesian methods have been specifically designed for concatenation. So they should not be used for a single protein tree. You should avoid to use them for a single phylogenetic tree. But now ML methods, they are new, like IQ3, they are very efficient ML methods. But the take-home message would be that the method is not the most important point. The most important point is the data set, scrutiny of the data set, because I have shown with the same method, if you change the protein present in the data set, you can get completely different results. So I know that during the recent year, people really focus on the method and say, we need better method, better method, better method, but they forget about looking carefully at the data set. And by the way, now in many papers, people publish concatenation, but they don't publish the individual tree. And then it's sometimes it's very difficult for you to recover the individual tree to see what's happened. And this I would recommend, highly recommend, to publish easily accessible single tree when you publish a concatenation.
Correct. I'll take this question, which is on concatenation, and the question is again anonymous. Considering that different concatenation of genes produce different trees, how can we select the right one?
I think to use a larger protein is the best thing, and also to see if the tree provide good, when you look at the universal tree, then you should look if inside the universal tree, the phylogeny of archaea, phylogeny of bacteria, phylogeny of eukaryote is okay. This would be an indication if the overall tree is correct. This is an important point. So if you use a protein, you produce a universal tree, and the tree inside the tree of archaea is very different from the consensus one, even if there is no perfect consensus one, but there is globally, we know that, let's say, Tomarchaea are close to Bettyarchaea and so on, then you have more chance to have a good tree.
Okay.
So,
yeah. The next question is from Hu Xiaojian. The question is, do you think the present study can confirm that eukaryotic cells originated from archaea? If not, what else proof do we need for this?
I don't understand the beginning of the question.
The question is, do you think that the data that you have presented can confirm that eukaryotes originated from archaea?
Yes, I think for me, they don't originate from archaea. They share common ancestor. And what we have shown also is that for eukaryogenesis, the coevolution of proto-eukaryotes with their viruses also play a very important role. Many proteins which are present in eukaryote and not in archaea came from viruses. Some people think that the nucleus also more or less came from viral nuclear factories, things like that. So we think that, in fact, when you want to try to understand the origin of eukaryote, you have to consider the protein they have in common with archaea, which were present in the last ancestor to both domain. But you see that you have also the introduction of bacterial protein via mitochondria or via other endosymbiont, and then you have introduction of protein from viruses. So this is very complicated and very exciting and interesting story.
Right. Aaron has added a piece of information, Aaron Oren from the ICSP. He's added a piece of information concerning the classification and nomenclature of the higher taxa that is above the level of phylum. And he says the following paper is now in press and proposing to include the nomenclature of the higher taxa in the ICNP. And the paper is now in press, a proposal to include the categories kingdom and domain in the International Code of Nomenclature of Prokaryotes.
Yeah.
So this is a new piece of information. So now with the proposal of phylum getting accepted and now kingdom and domain coming picture, I think we'll have a more stable system.
Okay.
Yeah.
You know that for viruses.
Yeah.
You know that for viruses, the International Committee for Viral Taxonomy now has proposed also new nomenclature for viruses, which is the same as for the cell, except for the domain, which is now the realm. So the equivalent of domain for viruses is a realm. The highest taxa is a realm.
Right, right. I'll take the last question. The last question is from Alessandro Costa. And the question is, I hear that you cite the last author every time, but the eventual authority is the first. Could you tell your opinion about?
Oh, I think it would be the best would be to mention both the first and the last, because of course the person who did the work should be acknowledged, like Anja Spong in the work of the 2015 paper. The people who direct the work who get eventually the idea at the beginning of the work should be also acknowledged. But people in between are also sometimes very important. Sometimes they are two first authors. So
Yeah.
I never really think when I give a talk about that. I change from sometimes I say that for the 2015 paper, sometimes I say Etema's team, sometimes I say Anja Spong.
Yeah.
It depends.
Yeah. Just for your, for our participants' information, Anya Spang will be our inaugural speaker for the 2023 lecture in February.
Okay.
So she is already scheduled to speak at BISMiS Live in February 2023. And another piece of information is many a times the last author is actually the principal investigator of the lab, because that's typically a common feature in the scientific fraternity, that the person who's contributed most in terms of wet lab and actual bioinformatics is typically the first, but the person who is supervising or guiding the research is usually the last person in the author list. Yeah, that's a norm. I also follow the same for my lab.
This is I follow the same for all my papers. I follow the same for all my papers. I have an amazing story, if you want, about that. It had a colleague a long time ago who liked to be first author. So she was working with a technician, and she put her name first, always the name of the technician last. And after 2 or 3 years, the American Society invited the technician to come for a meeting in the States.
Yeah. Yeah. I mean, it's really nice that people acknowledge each other's work, because science is a trust-driven field where acknowledgements are very, very essential, and it's important to do that. I think we don't have any more questions. Thank you, Patrick, for this enlightening session. Before we close, I would like to make a quick announcement about the next session. The next session will be by Luis Rodriguez, who is with the University of Innsbruck, and he'll be speaking on the C code registry in the month of November on the 19th. And I would like to take this opportunity to also share my screen once again and invite all the participants to become members of the Burgess International Society for Microbial Systematics. We are having a call for membership. So if microbes matter to you, then you join us. It's very, very important that we get our voice heard. And as an advocate in the field of microbial systematics, the society will be very happy to have more memberships for us. Thank you, Patrick, and thank you all the participants for joining today. We'll close our session here, and with this, thank you, everybody, and we hope to see you in the next one in November.
Thank you. Bye.
Thank you.